If you're like me and you're a neuroscience major and a minor in physiology where you only look at the human body, but in between there you have to do a biology paper, and the biology paper is about animals, and you're like, I don't want to learn about animals. And you're currently learning about the phylum body plans. I found it very uninteresting, but at the same time, you have to understand it. So we'll be talking about the 7 body plans of different phyla. Symmetry is if they can form mirror images. So if you think asymmetric, an example would be a periphera. Periphera is, if you've looked at pictures, they are in branch form, right? And they have no similarities between their body parts. And then there's radial symmetry. They don't have a left or right side, so if you just picture a circle or the jellyfish, Nigerians, I think, they don't have a left or right side, but they have a top and bottom. And then there's the bilaterals. Bilaterals are all other phylum, all the phylum that I learned. So an example would be chordates. If you picture a square, they can be split into left and right side and form mirror images. And then there's the tissue layers, so how many layers they have, how many tissue layers they have. And obviously this is in embryonic cell form, so when they're in the female body growing. So absent, they have no layers of tissue at all, so periphera. They have no layers. Diploblastic, they have an ectoderm and an endoderm. That's pretty much easy to understand if you just think that. Triploblastic, they have an endoderm and an ectoderm, and they have a third layer, which forms the cavity, which is called the mesoderm. Now, all these are formed in the embryonic cell, so if you want to watch a video on how fertilization or something occurs, they'll tell you. Body cavity, so if they have a space between the organs or not. There's 3 types. Acoelomate. Acoelomate, they have no space. Pseudocoelomate, their body cavity holds organs loosely in place. So if you think an ectoderm and an endoderm as one layer, and then the mesoderm as another layer in the middle, and between them there's a huge space. It's still held in place, but there's a huge space. I hope you understand that. And then there's coelomates, which are pretty much opposite of pseudocoelomates, so that their coelom hold the organs in place. And then segmentation, so absent or present or absent. So present, if you think of worms are grown in segments, right? So they shed, and once they shed, they keep growing. And if you think our digestive system, it keeps growing in segments. And then absent, there's no grown, they're not grown in segments, so they stop growing. And then skeleton type, skeleton, endoskeleton, hard-ended internal structure, skeleton. So pretty much us, we have an internal skeleton. An example is the periphera. Actually, periphera, they do have an endoskeleton. They have these little structures in them, and echinoderms and chordates. Exoskeleton, they have a hard covering that surround the animal. So an example would be mollusks, the snails and arthropods, the crayfish. Now hydrostatic, if you picture a balloon filled with water, the water creates a pressured environment in the layers, so that's what holds the compartments together, the pressured filled environment. And then gut type, so where does the food enter and exit? This is really easy to understand, but you can mislead it. So a one-way gut, the food enters on one opening, so the mouth, and exits in another opening, the anus. Now, this goes in one direction through our body, so through our digestive system. A two-way is when food goes in one opening and the waste goes out that same opening. And then the last is the coelom derivation. So if you've done reproductive anatomy, you should know how our coeloms are derived. So we are deuterosomes, and if you think deuterosomes, you'll think dry pocket anus. So if you remember this, you'll be sweet. So the D stands for deuterosomes, R stands for radial cleavage. Cleavage is when how the zygotes cleave. So deuterosomes cleave radially. Cleavage meaning pretty much mitosis, dividing. I equals the indeterminate. So the tissue layers formed organs. The tissue layers that form the organs are indeterminate in the embryo, so their fates haven't been decided. Pocket is the coelom and the deuterosomes form out pockets that vaginate out of that layer. So our coelom would form the mouth. And then anus, just meaning the blastopore forms the anus. Now for the protozoans, if you know dry pocket anus, it's pretty much the opposite. Yeah. Examples for deuterosomes would be chordates and annelids, protozomes, arthropods, and mollusks. And yeah, I hope this video helped, and good luck in your exams or tests. Thanks.